Buenos días a todos. Buenos días a todos. Bienvenidos a este curso corto, que es parte de las actividades de la cátedra Eugenio Méndez de Ocurro. Me voy a permitir hablar en inglés, cortesía a nuestro invitado. Así es que, bueno, good morning, everybody. Welcome to this short course, which is part of the Eugenio Méndez de Ocurro Professorship of the National Polytechnic Institute. I want to welcome our speaker, Dr. Werner Gellermann. Dr. Gellermann, it's an honor, and we are very pleased that you have accepted our invitation to participate in this event, which commemorates the 60th anniversary of the foundation of the Physics and Mathematics High Studies School of the National Polytechnic Institute. Welcome, and thank you very much for giving this today course to students and researchers of our institute. Welcome, and thank you very much for accepting. My name is Miguel Tufiño Velázquez. I am the director of the Physics and Mathematics faculty of the Polytechnic Institute. Well, in fact, we are commemorating 60th anniversary of the Physics and Mathematics School and the 85th anniversary of the National Polytechnic Institute. So for this reason, we have organized several events. One of these events is this short course that you are going to give us today and tomorrow. I know that you are in Germany. Communications are allow us to talk in spite of the very long distance. So, but it looked like as if you were close to us. Well, thank you. It's an honor to have you here. And I'm going to give I'm going to ask Jaime Ortiz, which was your mate when he studied when both of you studied in Utah. Jaime is my mate of the same generation, so we are very close friends. And it's an honor to meet you. So Jaime, please, if you want to read his biographical sketch, and let him start with today's course. So thanks a lot, Dr. Gellermann, again. Thank you very much.
You.
Welcome you. Welcome. We are welcome to you. Thanks a lot. Glad to meet you.
Nice meeting you. Thanks for inviting me and letting me talk about our technologies.
Yes, very interesting. Thank you very much. Please go ahead, Jaime.
Okay, Werner, I'm going to speak in Spanish for the audience. Tengo el gusto de presentarles al doctor Werner Gellermann, amigo y condiscípulo mío de hace casi 40 años, cuando ambos estudiamos el doctorado en física del estado sólido en la Universidad de Utah, compartiendo el mismo director de tesis. El doctor Gellermann estudió su licenciatura y maestría en el Instituto de Óptica Cuántica de la Universidad de Hannover, Alemania. Tiene alrededor de 81 artículos científicos publicados, 12 patentes y una marca registrada. Varias de sus patentes involucran el desarrollo de nuevos láseres de estado sólido en el infrarrojo cercano y el uso de estos y de otros láseres en la investigación básica de las propiedades ópticas de cristales iónicos, polímeros conjugados y otros materiales de estado sólido. Además, en colaboración estrecha con investigadores médicos durante los últimos 24 años, ha investigado metodologías de detección óptica no invasiva para carotenoides y compuestos similares en tejidos humanos vivos y ha desarrollado nuevos instrumentos alrededor de esta línea de trabajo en óptica biomédica. A lo largo de su carrera académica inició varias empresas y desarrolló tecnología patentada para detección de carotenoides cutáneos para aplicaciones en la industria de suplementos nutricionales. Desde 2014 se dedica a tiempo completo en la administración de 2 empresas comercializando sus patentes en imagenología de pigmento macular y en el desarrollo de nuevas tecnologías de detección de carotenoides cutáneos. Las 2 pláticas que nos dará el doctor Gellermann hoy y mañana las hará desde Alemania, donde ahora se encuentra temporalmente atendiendo a su octogenaria madre, por lo cual me ha pedido que el número de preguntas al final se limiten solo a 3. Sin embargo, me ha comentado que pueden interrumpirlo para hacer preguntas en cualquier momento durante su plática. Okay, Werner, now the time is all yours. You can please start your talk, please.
Okay, let me see whether we can share the screen here. Okay, do you see something?
Yeah.
Good.
Okay, great. Well, thank you very much again for letting me talk about our technologies. This is some work I have been doing for the last 20, 25 years or so in collaboration with various medical folks. So originally my education was in solid state physics and spectroscopy, and I decided to kind of apply those technologies to the field of biomedical optics. The title of my talk is Non-invasive Optical Detection of Carotenoids in Living Human Tissue. The motivation here is as follows. Large-scale epidemiological studies have shown that antioxidants like carotenoids play an important protective role for the health of the human body. So important protective role is the key word here. And the question is it possible to detect those carotenoids in a non-invasive optical way in those living tissues? And then if yes, then we would have a novel in vivo tissue diagnostics possibility, which would allow us to screen and track those antioxidant levels in large populations. And then we could maybe monitor or assess the risk for degenerative diseases with a quick optical test. So the outline of my talk is as follows. I'm gonna list a few properties of the carotenoids, then kind of sketch the oxy and antioxidative processes in tissue, and then talk about our optical detection methods and applications in some medical disciplines. By the way, if you like, you can at any time interrupt me also with questions. I'd be happy to take questions as we go along. So what are carotenoids? You have all seen them. Here's a picture of fruits and vegetables. And it turns out that all of these bright colors here, like the red color in strawberries and tomatoes, the yellow color in squash, the orange color in carrots, they come from this large family of molecules called carotenoids. Obviously, they got their name from the carrots. And these molecules are taken up through the diet, and they function as efficient antioxidants in our various tissues in the body, and sometimes work as optical filters as well. And people have established that they protect our cells and cellular DNA, so they play a very important protective role. These molecules are here shown, the major ones are shown on the right-hand side, and you can see that they all are long chain-like molecules consisting of alternating conjugated carbon-hydrogen bonds. So this is a sequence of single and double bonded carbon here, and then you have hydrogen dangling off to the side. in some ways is kind of resembles a metal wire where the electrons can move very rapidly along these little wires here. And then depending on what kind of carotenoid you have, whether it's beta-carotene, lycopene, zeaxanthine, lutein, beta-cryptoxanthine, phytoene or phytofluorine, you see that they all look very similar, but they have different end groups here. Yeah, in the term of beta-carotene, there's one group here, one group there. For lycopene, there's no group. Yeah, just a chain. For lycopene, for zeaxanthine, you have a ring here at the end again, just like in the beta-carotene case, but there's some additional OH molecules hanging off of the ring. And so on. So they all differ just a little bit, but overall, they generate pretty much as conjugated, what we call conjugated pi electron conjugated molecules. And since they're pi electron conjugated, the electrons, again, they can zip along these wires very efficiently, and that leads to very strong optical absorptions. It turns out that they all absorb very efficiently in the blue-green wavelength region. So since the tomatoes look red here, yeah, they absorb in the blue. And then other carotenoids, like beta-carotene here, they're a little bit shifted, but not much. So they're shifted from red to orange, but pretty much all absorb in the blue wavelength region. Okay, so why are they really important? So here's a slide that kind of shows the relevance of them in medicine. One example is the case of age-related macular degeneration, AMD. That's how I got introduced into that field. I met a retinal surgeon at our eye clinic at the University of Utah, and he challenged me to come up with optical technologies that can detect this spot here in the human retina, right in the middle of the macula, which is generated by lutein and zeaxanthine carotenoid molecules. That's where we started. I'm going to talk about that particular topic tomorrow. Here's another example where people in Baltimore, at the University of Baltimore, a long time ago, 2006, have conducted a study where they tracked blood carotenoid levels and also collected epidemiology data, and they tracked the all-cause mortality. Yeah? So they started with X numbers of people at time 0, and then they tracked the people over 60 months, which is 5 years. And they realized that when you look at the subject's carotenoid levels, the highest quartile in carotenoids, which is this red curve here, has a larger survival rate. So after 5 years, people with high carotenoid levels lived longer than people with low carotenoid levels. Yeah, the lowest quartile is shown here. So there's a significant, maybe 10% difference between subjects with high and low carotenoid levels if you track them over 5 years, and if you look at their survival rate, yeah, including all kinds of diseases, degenerative diseases, cancers, what have you, everything. So that gave people kind of a epidemiological piece of relevance for these carotenoids, and that's our motivation. That's why we got started. Some diseases here are age-related macular degeneration, cataracts in the eye, which is a breakdown of the natural lens, premature skin aging from bombarding the skin with ultraviolet light, a lot of sun exposure, especially at high altitudes, then some photosensitivity disorders, and there seems to be also a correlation with certain forms of skin cancers like basal cell and squamous cell carcinoma. And then also I'll talk about this tomorrow. There's a application for the retinopathy of prematurity. It's a big word, just meaning that children get also affected by low levels of carotenoids. Okay, let me show you some examples of tissue carotenoids. One example that I like right now here, for example, when I look out of the window here in Germany right now, you see that the green leaves all have turned colors because of the chlorophyll has died off. And what's left behind are these brown, orange, and magenta colors here, yellow colors in the same leaves. And all of these colors are generated by carotenoids. And they play a very important protective role, actually, also in the leaf. Here is an energy level scheme showing for the carotenoids. You have the ground state level, the singlet excited level, and the second excited level. And it turns out that the energy spacing here for the carotenoid molecule matches the energy spacing of the chlorophyll molecule that you have in the leaf. The chlorophyll molecule is known to, of course, as you know, it's an extremely important light harvesting pigment. Without photosynthesis, the life on Earth would be impossible. But it turns out that chlorophyll has some bottleneck in its optical excitation, and carotenoids sitting right next to them help them to quench this bottleneck. So this allows the chlorophyll molecule to cycle many times repeatedly through the light harvesting process. So very important function also in biology, in botanics. Here's another example, carotenoids in lobsters, which you might like to eat. Here's a lobster in its natural form. It has a purple shell. And then when you boil it, you probably have all seen this here, it turns bright orange, orange red. So a big color change from here to there. And what happens there is that the carotenoid molecules, which sit in the lobster shell inside some complicated protein pocket, when you boil it, the carotenoid molecules come out. And then the residual configuration here is separated, and you see the color of the carotenoids, which is bright orange again. The people understand these carotenoid protein structures quite well now. It's a very complicated process done with supercomputers. What people do is they grow a protein crystal first and then do X-ray diffraction spectroscopy, where you have this blast some X-rays onto these protein crystals, and then you analyze with the help of computers the composition of the protein molecule and understand where the carotenoid molecules are situated in those complexes. Okay, now let me talk a little bit about oxidative and antioxidative processes. You all have seen, if you cut a pear into two pieces and let it sit at room temperature, you see that here, starting from its white appearance, at the beginning, after about half an hour, it turns brown, okay? And that's a classical oxidation process of tissue. It's a biological tissue, and the pear goes from white to looking some brownish color. And that's because there is a slow but possible chemical reaction of the normal oxygen molecule, which is a molecule, and it's very unique in nature because in its ground state, it has a triplet state. It has a triplet ground state, which means that in its state here, in its second orbital here, it has unpaired electrons, okay? And in chemical reactions, you can only have reactions between molecules of the same parity, gerade and gerade molecules, or ungrade and ungrade. So not usually between ungerade and gerade, okay? But that's what's happening with the oxygen molecule. There can be a slow chemical reaction between the triplet ground state of the molecule and the singlet ground state of the organic other tissue molecules, yeah, sitting in the pair. So there's a slow reaction between triplets and singlets possible in this biological tissues. Actually, this is a very interesting process, and this oxidation is actually not always bad. In medicine, for example, it is used on purpose sometimes in a process called photodynamic therapy, and that works as follows. What you do is you inject a sensitizer into the bloodstream of the people, and then activate it with light. You shine white light on it or laser light. And then what happens is that the ground state sensitizer molecule goes through its excitation and goes into a triplet state, in its excited state. And in that triplet state, it can react very efficiently with the ground state oxygen molecule, which, as you remember, is in a triplet state as well. So you can have very efficient reaction here, and then you create this singlet oxygen molecule, which is depicted here as a little bomb, because what it does is it reacts very efficiently with the surrounding neighbors, much more efficiently than what you saw in the pair, where the reaction took half an hour. So if you inject this process, photodynamic therapy, is used, for example, in eye surgery, where if patients have some abnormal blood vessels in their retina, you inject the sensitizer and then shine light onto these growth, and then you can destroy, through this activation process and the reaction between the singlet oxygen molecules and the ground state tissue molecules within a very short time. Here are the numbers. Within 1 minute exposure time, you can achieve a penetration depth into the tissue for about 3 millimeters. And then the sensitizer molecules are slowly flushed out of the patient, which has to be kept out of light for several days. So very important application of this very highly reacting chemical process in medicine. Okay, but for our purposes, we don't like these oxidative processes, and we explore the carotenoids as antioxidants, okay? So it's very simple. So here you have again the oxidative cartoon that I showed you before, leading to all of these damages here with the tissue. And what you do is here you bring these yellow carotenoids in close neighborhood of the singlet oxygen molecule in case it forms. And that will block it because there's a very efficient reaction of the carotenoid molecule with a singlet oxygen molecule. And this is called an energy exchange reaction, where the singlet oxygen molecule in excited state reacts with the ground state carotenoid molecule such that the singlet oxygen molecule is deactivated. It goes into its ground state, and the carotenoid molecule takes up the energy and gets excited. Yeah, so you transfer the energy from here to here. And then you have a harmless oxygen molecule again. And the excited carotenoid molecule, all that it's doing, it's just starting to vibrate a little bit along its carbon backbone chain, and then just dissipates the energy that way. Okay, so this is kind of the background information regarding oxidation, antioxidative effects, and processes playing a role in tissue. Okay, now let me talk about the optical properties of carotenoids. Let me switch this here first into presentation mode. In the ground state, we have the carotenoid molecule here with its vibronic substates shown here in the electronic ground state configuration. And then when we excite the molecule here, for example, by shining blue light onto it or white light where it fishes out the blue light components from the light, then you see a very strong optical absorption. And that absorption goes from about 350 nanometers to about 500 nanometers. So it's a very broad range, and it happens in the blue wavelengths region of the spectrum. And it's very strong due to the pi conjugation of the electrons zipping up and down the chain. And what you see here, these peaks, these narrow bands, those are vibronic bands coming from the transitions from the ground state projecting here onto the vibronic substates of the in the excited state configuration. Okay, so that's kind of a typical carotenoid spectrum. And then what happens is this carotenoid molecule relaxes very quickly within 100 femtoseconds or so to some other excited state. It's a very well, not too unusual. It happens actually in several configurations, also in solid states. Dr. Ortiz knows very well about this. Actually, it happens in some materials that we studied a long time ago, which are called color centers, F centers in alkyl halides, where you have a similar process where in the excited state, the color center also is in a Grade state. So we have a Grade state in the excited state as well. And then what happens is that you have a luminescence occurring from this state. And since the luminescence happens between a Grade state and a Grade state, according to the laws of quantum mechanics, is actually forbidden. That means it's very weak. And if you crank up your detector and arrange a high gain, you can still see the fluorescence, and the shape looks like this. It occurs here in the peak of about 530 nanometers, with a relatively small Stokes shift. So 530 is here next to the absorption. And you see also in the emission, you see some vibronic substructure. But again, the emission is extremely weak. Okay, the absence of fluorescence here makes it possible to explore some spectroscopy, which is named after the physicist Raman. So it's Raman Spectroscopy, where you can excite the molecules from the ground state here, this blue arrow, into the excited state. And then before any relaxation can occur, the molecules comes back down to the ground state, okay? And you can do this in the absence of fluorescence because this fluorescence is very weak. So that's why we decided as a first attempt to use Raman spectroscopy for detecting these carotenoids in living human tissue. So here's what we do. We take a laser beam and a blue laser beam and shine it into a cuvette, which is filled with carotenoid molecules, these chain-like molecules like beta-carotene. And then we look for the Raman scattered light, which comes out here to the side. And it's governed by some equation here, which basically shows that the scattered light intensity IS is proportional to the excitation light. And then there is some Raman scattering cross-section, which in general is very complicated, depends on the symmetry of the molecule, all kinds of stuff. But it's not, we don't need to go into that. And the last proportionality factor here is N, which is the concentration. So it's a very nice spectroscopy where the scattered light scales up linearly with the concentration N of the scatterers that you have sitting in the cuvette. Okay, so then you can kind of get a handle on the concentration of the molecules through the scattered light intensity. Okay, here's a typical Raman spectrum of these carotenoids, and what it looks like is you see very strong pronounced peaks here. And one is at 1500 wave numbers, which is roughly 20 nanometers away from the excitation light. And there's another one here a little bit closer. It's another very sharp peak, which belongs to the carbon-carbon single bonds. This peak here belongs to the double bonds. And then this peak here belongs to the methyl side groups that are dangling off the molecules along its backbone here. So very interesting, precise spectral signature of these molecules, okay? So then what we can do is we can shine the laser beam onto the tissue and look and try to see whether we can detect these sharp carotenoid peaks also coming from the tissue. Okay, here's again the highlighted the proportionality to N, okay? Okay, here's what happens to a spinach leaf. If we take a spinach leaf from the grocery store, take it to the lab, expose it with a laser beam, then you see that the Raman spectrum here contains the characteristic 3 peaks from the methyl, the single bonds, and the double bonds. So it works very well even through chlorophyll. There's some fluorescence background here coming from all kinds of other organic molecules in the tissue, but as you can see, there's a possibility to discover those carotenoid peaks. All right, so we did this then with a bunch of carotenoid solutions. Here are all their absorption spectra shown on the left-hand side. Yeah, here's beta-carotene and its absorption band. Here's zeaxanthin, here's lycopene, here's lutein, here's phytofluorine, and you can see in most cases, these carotenoid bands here occur pretty much the same, in the same wavelength region at around 450 nanometers in solution. And here are their Raman peaks. You see there's almost no difference in the spectral position of those peaks here except for phytofluorine. Yeah, but this one, zeaxanthin, beta-carotene, they all occur pretty much at the same spectral position. Okay, if we want to deal with living human tissue, then we cannot just shine a laser beam onto the tissue and declare victory if we see the carotenoid peaks, because the Institute of Medicine tells us that the only acceptable method right now, before we started, are measurements of the carotenoids in blood. Yeah, so blood concentrations. They're considered the best biological markers for the existence of carotenoids and then also for the consumption of fruit and vegetables. So if we want to convince the doctors that our optical technologies work, then we need to convince them that we have a good correlation with the gold standard of blood measurements. Of course, blood measurements are not very attractive in medicine because they are invasive. A lot of people don't like the pain associated with the blood extraction. They're expensive to analyze. And of course, it's also quite time consuming before you get the results coming back from the lab. So, you know, for many medical applications, the blood measurements in large populations are sort of prohibitive. Okay, now if we want to develop optical skin carotenoid detection methods as a simple non-invasive alternative, we need to look for correlations between the carotenoids in blood and the carotenoids in the tissue. To give you an idea what it looks like in blood, here's what we call a Hplc chromatogram, which is high pressure or high performance liquid chromatography. Basically what you do is you take the blood, you spin out the hemoglobin, and then you squeeze the serum, what is left over. You squeeze that through a fractionating column at high pressure, and then you look for the molecules coming out at the other end of the column, and you analyze them with optical absorption spectroscopy coming out. And then you will see that the smaller molecules come out first here on the left side, lutein, zeaxanthin, and then the larger molecules come out a little bit later. Okay, and biochemists know exactly the time points when these carotenoids come out. So if you have a peak here appearing after a few minutes, let's say here about 3 minutes, then you know it's lutein. If it comes, this peak here comes a little bit later, maybe that's alpha and beta cryptoxanthin. So that's how HPLC works. Now it turns out that all carotenoids found in blood also appear in human skin. So that's very good. So now we can target to measure those carotenoids also in human skin. So what does a human skin look like? Here's the skin layer system. We have on the surface of the skin, just below the surface, we have a layer which is pretty much a scattering layer called the stratum corneum. Okay, that's a lipophilic layer. That means it's fatty tissue, and that's a few hundred micrometers thick. And below that, you have the epidermis here. It's called the epidermis. And at the bottom here of the epidermis, you have some basal cells which grow out towards the surface. And every day, the human body sloughs off some of these semi-dead cells here at the surface, and the new cells grow up again out of the basal cell layer towards the top. And so it's kind of a dynamic situation. And the carotenoids exist everywhere here in the dermis, in the epidermis, in the stratum corneum. And but with light, if we come with light excitation here from the surface, we don't have a chance to go through the whole tissue layer system, but we have a good chance here to penetrate a little bit into the stratum corneum, maybe a little bit here into the epidermis, and we can look for the scattered light coming back out of this tissue. Okay, so it's the optical method would be restricted to measurements near the surface, but you know, we have a good chance to catch whatever exists in here in terms of the carotenoids. Okay, one thing we have to consider in living skin is when you look at your skin, you see it doesn't look just yellow, but actually it everybody's skin is a composition of basically three pigments, three photopigments. One is beta carotene here, which and other carotenoids which come from the diet and are deposited into your skin. The other one is blood, yeah, oxygen, hemoglobin and deoxyhemoglobin, heme protein. They generate a very strong color here in the 500 to 600 nanometer range. Yeah, it has very strong optical absorption. This is not shown to scale here. Yeah, this would be very strong. Beta carotene would be a little bit smaller. This is just highlighted here to show again the vibronic substructures here. And then superimposed on all of these chromophores here is also melanin, which is a very large protein molecule that generates a scattering and color all the way from the near red to the ultraviolet. So this generates a brownish color, also scatters, and all of these other chromophores are superimposed on this, okay? Okay, here's our first attempt to detect carotenoids with resonance Raman spectroscopy. Resonance is because we shine the laser beam directly into the optical absorption band of the carotenoids in the blue wavelength region. And in our first attempt, we used 488 nanometer light. That's in the blue. In the beginning, we used an argon laser here. And so shining directly into the strong optical absorption bands of all of these carotenoids into the skin. And then we look for the Raman spectrum coming back out. As a typical tissue site, we use the palm of the hand or the finger, and the measurement is about 30 seconds. And then what you get then is this here, shown at the right side here of the computer monitor. Here you have the raw Raman spectrum. Yeah, it's a very strong fluorescence background. And if you don't look carefully, you don't see anything writing on top. But if you amplify the spectrum here, do some background subtraction, then you can retrieve here the spectrum shown on the right-hand side. And this is the typical spectrum for the carotenoids: methyl band, carbon single band, and carbon-carbon double band, okay? So we can identify these peaks coming from the skin. Here's another lab prototype shown where in the beginning we used an argon laser a long time ago, but more recently we switched to a solid state laser, which came on board, which was invented by people in Japan. That's hidden in here. And that offers us for the first time the possibility to generate a lab setup that's portable. As you can imagine, an argon laser is not very nice to carry around, but the solid state laser here, which exists in the blue, does the job, and we can use it to develop a portable instrument. These are our first results. We scanned about 1,000 people here, and what you see here is a histogram of carotenoid intensities derived from the Raman spectra. So people with a high Raman peak have a high score, and people with a low carotenoid peak have a low score. And then if you plot all of the data for all of these people as a function of the signal strength here, then you see you have some people with a very low score, a very low number also of people with a very high score, and then most of the people with some medium score here peaking at about 20,000 Raman units here. Yeah, so this is a classic histogram, and as you can see here, it's a kind of a nice bell-shaped curve, where when you look at that and you get measured and you happen to be here, you can be happy because you have a high carotenoid level. If you're here in this range, you're medium, and then if you score here in this range, you would consider to have low carotenoid levels. So these first lab results kind of showed us the feasibility to assess carotenoid contents in large numbers of subjects. Here we scanned 1,000 people roughly, and we can do it very quickly. Each measurement takes about 20 seconds. And now we can use this technology in clinical trials to study the correlation between disease and tissue carotenoid levels. Also, we can look at the bioavailability of the carotenoids. We can look at the correlation with blood levels. We can look at oxidative stress in various tissues and so on. Okay, now I come to the topic of validation studies. Very important. Initially, I thought, well, if I show these medical doctors our technology, they will like it. But the first thing they pressed is that you need to show us that this is really a valid technology. They are only used to blood measurements. So here we come with an optical technology and claim it works. So we need to convince them that it really works in their mind. So we work together with various medical disciplines, and first established a correlation between our optical carotenoid scores and carotenoid levels in serum as determined by Hplc. Here's a first result taken for about 100 healthy male and female adults. Optically, we used the inner palm. Blood was drawn, of course, from the arm. And what you see here is along this axis, you see the total serum content of the carotenoids, which is measured in micrograms of carotenoids per milliliter of liquid. And then here on this axis here, we have our Raman score of the carotenoids. And what you can see here is on average, we have a very decent linear relationship where if you have high levels here of total blood carotenoid contents, you also have high Raman response. And if you have low serum levels, you have low Raman response. So the correlation coefficient here is about 0.78, which is considered very nice in statistics in medicine. So that looked very promising. Okay, then next step was to look for validation also not just with blood, but also with skin, because you can argue that the skin has different properties compared to the blood. Yeah, maybe not all molecules go in there, whatnot. So we quickly scanned amongst ourselves the carotenoid response with Raman spectroscopy from our heels, the heel of the foot. The heel of the foot has some very thick stratum corneum, and it's a convenient site to use our optical probe here. And then what we did is we cut off some thick stratum corneum tissue here with a razor blade and analyzed it with Hplc. So we ground it up, analyzed it, and it looked like we have also with this excised tissue here, we have a decent linear relationship. Actually, with this excised tissue now from the analysis, we can establish a direct correlation between the Raman signal and the concentration in the tissue. By the way, can you still hear me?
Yes, I can hear you. Somebody, ¿alguien puede, por favor, apagar su micrófono? ¿O están preguntando?
Okay, so the connection is okay?
Yeah, I can hear you very well. And
okay,
see,
great. Then I'll just go ahead. Is there a question or?
I'm trying to figure out, but I think somebody left his microphone open. Okay, just go ahead, please.
Okay, then we teamed up with a group at Yale University. There's a famous epidemiologist, Susan Main, and her co-worker, Brenda Cartmel. They are very well established in the field of epidemiology. We're very interested in our technology. And they also independently verified that there is a strong correlation between skin biopsy and plasma with the Raman spectroscopy. Also, they looked at a correlation with food and vegetable uptake, which is measured in the field of nutrition through questionnaires. So just ask people what they eat. It's actually quite a sophisticated questioning process. And then they can look at correlations, and there was also a decent correlation. So that was very encouraging as well. One possibility that we can have with the use of lasers is we can play some games where we use lasers with different colors. We use a laser with a blue color and a laser with a green color, and can then see whether we can measure some of these carotenoids selectively. Yeah? Can we, for example, tell the difference between beta carotene and lycopene in the human skin? Because as you remember, maybe if you paid close attention, the beta carotene and the lycopene are slightly spectrally shifted. So if you use strategically blue and green laser wavelengths, maybe you can divvy them apart. So that's what we did here with this laser, where we could switch from blue to green and then sequentially measure the Raman spectra of people's tissue. Okay, that's shown here. Here's blue and green. As you see, the green excitation is much lower than the blue excitation. And if we do the algebra right, we can divvy apart the lycopene versus the beta carotene. I don't want to go into the equations here, but the end result is shown here at the bottom. Basically, the black bars and the gray bars here depict the strength of the concentrations in the body, lycopene versus beta carotene. And what's interesting in here, you see that it varies in people. It's not always the same ratio between lycopene and beta carotene. But if you compare this person here, for example, with this person here, it's reversed. So here, the beta carotene has higher than the lycopene, and here it's the other way around. So that's a consequence of the diet. What's very interesting is in the field of medicine is there's a very strong interest in lycopene. Lycopene is considered to be beneficial in preventing prostate cancer. So, that's one of the reasons we did this, to see whether we can selectively measure lycopene. And what we did for some of our guys is in the lab, we supplemented ourselves with lycopene. And as you can see here, over time, the lycopene levels go up with time. Yeah, we can monitor this. Whereas here, using beta carotene instead, the levels stay the same. So this might be a way in the future to track lycopene levels in the skin and then correlate it with disease progression. Okay, now let me show you some applications of the technology in various fields of medicine, in nutrition science, in neonatology, orthopedics, and then in the end, in the nutritional supplement industry. Okay, first example here is a controlled dietary intervention and observational study carried out with collaborators from the USDA lab in Grand Forks, North Dakota. They have a government lab there dealing with nutrition, and they have a very strong interest in these optical methods. So they try to check and see whether you could really manipulate the diets and see whether the manipulations correlate with the optical measurements. So here's their study. Here's the advertisement, get paid to veg out. So they recruited some students and other volunteers from their lab and paid them $1,000 per head and asked them to get involved in a study that includes 4 phases, lasting up to 8 weeks, about 2 months, where normally they would eat a normal diet in the first 6 weeks, normal diet, while excluding most vegetables and fruits. Then they would eat a healthy diet with high concentrations of vegetables and fruits. Then they would go back to the normal diet. And then the normal diet while excluding most vegetables and fruits. And then at the end, they would return to their normal diet without any restrictions for 8 weeks. Okay, so they enrolled women and men at aged 18 to 65. No constrictions on weight, but they all had to be non-smokers. Okay, here again, the 1, 2, 3, 4 phases of their study. The first phase called a depletion study. Second phase, a high carotenoid feeding study, a diet fortified with carotenoids. Then the third phase, a depletion study. And then the fourth phase, going back to their natural diet. Okay, and then here's what happens. Here are the results from the Raman measurements. You can see here in the first phase, everybody's levels go down. In the second phase, most people's carotenoid levels go up. And then here in the depletion phase, they go down again, and then at the end, they level out. So they did the statistics on that and determined that it works acceptably well for the field of nutrition science. So they helped us establish this as a valid and reliable biomarker of change in vegetable and food consumption. Okay. And another thing they saw is that the optical measurements can be used to assess the effectiveness of high vegetable and food interventions as quickly as within 2 weeks. So that's how long it takes for the carotenoids to go from the vegetables into the bloodstream and then into the various tissues like the skin, about 2 weeks. Okay, next application here was with the University of California in Davis in their Department of Nutrition. They wanted to look at a dietary intervention in school children, in particular 4th and 5th graders. And they wanted to see how, what could be done there in that cohort. So here are all of the participants. They recruited 171 children and measured their Raman scores optically. And here you see the histogram again. So most children had a score here between 20 and 30,000. Some kids very high scores, some kids low scores, but the mean was about 30,000, okay? And then what they did is they conducted a teaching program where they taught the kids for 1 semester about the advantages and protective effects of carotenoids and educated them in the importance of a healthy diet, okay, for about half a year, 1 semester. At the start, the average here was about 8.9 on this scale before the intervention. And then after all of these teachings, you see the levels drop down to 6.8. So that was a very disappointing result for them because on average, the levels in these children went from whatever level they had before, it went down. It went down by about 20% or so. And this is not due to the optical technique. You could argue, well, maybe the Raman system doesn't work, but they tracked the depletion also in the blood of the children. They incentivized them by giving them some monetary rewards and also drew their blood and showed that their blood levels are depleted as well from before to after. So for whatever reasons, the levels went from a high average state to a lower state. And it's confirmed by blood measurements. And it shows that if you run an intervention study or something like that, it doesn't mean that it's going to be very effective. Maybe people lose their interest in it, they become bored, who knows what. It's very tricky to change people's habits. Excuse me. All right, now a third example here is skin carotenoid screening in childcare centers. Here we collaborated with a childcare health program in San Francisco, Department of Public Health. This project was run by Jody Stuckey. She is an epidemiologist, and she told me that there's a very strong need to measure and track people in daycare centers, in child daycare centers. A lot of people are in these daycare centers spending quite a bit of time there, maybe up to 8 hours a day or so. And it turns out that at lunchtime or so, they get fed a relatively poor diet. So there is a likelihood that these children here in these daycare centers may be subject to a poor diet. So there's some interest in scanning these people in these daycare centers and then work with these daycare centers to improve the diet. So here's a result of one daycare center in the Mission District in San Francisco. You see here, maybe this a few hundred people You see a nice histogram again with the Raman peak here, 400 on this scale. Looks quite nice. And here you see the results of many daycare centers measured in San Francisco. Let's see, I think about 24 childcare centers. So they did this over the course of about 1 year. And here they rank the daycare centers depending on the average score of these children in the particular daycare center. So they have a cutoff point where if the child scores below 300 on the Raman scale, then they calculate that percentage for this particular daycare center and plot it here. So this daycare center, only 7% of the children were low. And in this daycare center, 80% of the children had a score below 300, the low score. So in this group here of daycare centers, about 50% of the children had a relatively poor diet. So they can now work with these daycare centers here. They know where they are and know the people involved and launch some education programs. One thing that was quite nice here, we took advantage of the situation in San Francisco. We have a very high ethnic diversity in San Francisco and use the subjects there to look at the Raman scattering coming from people's hands because the melanin levels, they differ quite a bit between ethnicities. Yeah, so there's a gradual increase here, probably from here to here, to here, to here, to here, and so on. So we wanted to see whether the Raman spectroscopy is influenced by the melanin levels. For that purpose, we used a independent commercial melanin meter. It's also based on optics and only measures the melanin levels with an optical method. It's produced by Konica Minolta. I don't want to go into all of these details right now, but you can figure out the melanin levels. Basically, the blackness of the skin measured in the near infrared. Okay, and then what we did is we scanned many subjects, 160 children, in this case here, as you can see, that as a function of the melanin index here, which we plot along the horizontal axis, we see that the Raman scores, they can go high or low independent of the melanin index. So here, most people had a melanin index of 0.8, and the Raman score can be low, can be medium, can be high. Yeah? So each data point here represents one measurement of a child. And you can see that it can be low or high, but that on average, there's a straight line here, which statistically means that the melanin index is not a factor. We get the same high or low reading independent of the melanin index. For example, if somebody here has a very high melanin index, a black child, for example, the Raman score can be as low as 50 here or as high as 700. Huge difference. So melanin does not play a role. And that was very nice for us because that's how we had adjusted our algorithm. Okay, here's a example from Neonatal Medicine. There's a strong interest also in this field because it turns out that prematurely born babies often are born prematurely because they have a strong oxidative processes happening in their bodies. And people would like to know, maybe assess non-invasively the carotenoid antioxidant levels in these prematurely born infants and also in their mothers to see whether they can establish a correlation between the lack of oxidants and the lack of antioxidants and prematurity of birth. And also it turns out that if a baby is born prematurely, then they have a very high risk for infections, for chronic lung disorders, for retinal problems, and so on. So it's a very interesting field of medicine where maybe our technology could help. So you see here we scan the heel of prematurely born baby. And one thing we had to do is we had to really tweak up our machine, because in general, the carotenoid levels of these preterm newborns are very low. See here about the 2,700 range, whereas in adults it's about here. So maybe 10 times lower, okay? So here you have preterm newborns, term newborns, infants, and adults. So the carotenoid levels go up. Here's a correlation between the skin carotenoid scores measured in these infants and their serum levels. Yeah, we have drawn blood from these kids. And as you can see here, we are getting, again, a very nice linear correlation. So it works as well. These correlation studies we have to do in each discipline of medicine, because each physician in that particular area wants to be convinced that the optical techniques work in their field. So they don't believe any other results. They just want the demonstrations for their patients. Here's some measurement of the carotenoid concentrations over weight. The weight of the baby goes up, so does the carotenoids. So it's kind of normal development, as you can imagine. And then here's a quick survey of what happens when the Raman data are taken for babies having different gestation weeks. You see if the baby has a very low gestation of 28 weeks or so, Raman levels are even lower. And then if the gestation weeks go up, the levels go up. So for the baby, it's more advantageous to have a high gestation week number, of course, not be born prematurely, because the carotenoid levels are up then. What's interesting is that for these physicians is also not just to measure the prematurely born babies, but also to measure the expectant pregnant mothers. And the reason is that the carotenoids in the baby are transferred in the fetus stage, and they're transferred from the mother to the baby, to the fetus through the placenta. And that leads to some depletion effects in mothers. So it's a very interesting medical challenge to improve the diet of expectant mothers, because besides iron supplementation, they do not receive any other supplements during pregnancy. And very recently, we managed to finally, after about 6 years, to get a new clinical research study funded from the NIH, looking at pregnant mothers and newborns, measuring their tissue levels, blood levels, and so on, and looking at correlations in a larger patient cohort.
Werner,
yeah,
there's a question in the chat. If you want, I can read it. It's a question from Gabriela Araujo, and the question is, if the part of the skin suffer an increment of temperature, it's the zone where the scan starts to capture the spectrum from the device. Is there some perturbation of in the spectrum by the increase of temperature, by the excitation that you use in the instrument?
Yeah, that's an interesting question. We looked at that in the beginning, but there's no effect from the temperature.
Okay.
So we cooled our hands. We put some ice on it. We heated it up a little bit, but there's no effect from that. Actually, the carotenoid molecules are very stable in terms of temperature. When you cook tomatoes, for example, the carotenoids stay intact there up to boiling temperature. or as you saw earlier, also in the lobster. So the carotenoid molecule by itself is very stable. It does not degrade with temperature. And of course, in living human tissue, the temperature range is not that high anyway. And in these prematurely born babies there, or babies or whoever, the temperature in the incubators are very pretty much controlled in a stable temperature region. But yeah, good question. We convinced ourselves too in the beginning about these effects. Similarly, also we looked at pressure effects. Does the pressure make a difference, or washing the hand, hydration? Hydration was actually an interesting question, because you could argue that the skin would swell up with hydration, but it turns out that it's not a decisive influence.
Okay.
All right, now another application area is carotenoids in bone health. And here was approached by a local orthopedic doctor. He's an orthopedic surgeon, specializing in total knee replacement. And he told me that when he looks at people's knee bones during surgery, that there are some huge differences in the color. Some people have a yellow color of the bone marrow, and other people have a very pale coloration, especially those people with poor nutrition. And it turns out that if you excise a bone from his patients and extract the bone marrow and run it through a fractionating column, you see here that in the bone marrow, you have all of the carotenoid peaks that you also have in blood. Okay. So it's really likely that those carotenoids, that those yellow coloration in those bones come from carotenoids. And then he asked us to show that optically. So what we did is, here you see some bone during surgery. They cut away the knee. This is the lower portion of the leg here. This is the upper portion. And you see here, this bone here is very yellow looking, and the bone here at the bottom is very pale looking. And when we do the Raman spectra of these two bones, we had some samples. Then you see here, there's barely a peak visible here, barely a peak visible here, and it's much stronger in the other bone. So we did this for a bunch of biopsied bones here, and see that indeed, in many subjects here, the levels can vary quite a bit. In this project, we are developing, and it's been taken quite some time now, a technology where we can measure those carotenoids, not with Raman through excised bones, but where we can directly go into the surgery room and measure the tissue surface, which would be here, and shine a light onto this tissue surface, and then measure the light which is reflected back over here. Then with the help of filter wheels, take out the difference between the yellow coloration and the background coloration, and quantify the carotenoids this way. So this project, we have not been able to secure some NIH grant, but the preliminary measurements sound quite interesting, because here we can also say that we can measure these carotenoids in the knee, and maybe we can use indirect bone carotenoid assessments through skin measurements. Yeah, we need to study that. Maybe what you have in skin manifests also what you have in the bone. And then you can have some nutritional intervention and hope to increase your bone levels that way, and have your knee joints and then last much longer, and also improve the healing. Turns out that the healing time is very long in patients with low bone carotenoid levels. So that might be also an interesting research area for orthopedic people. Okay, now let me come to By the way, do you see this taskbar at the top? Do you see the slide without the taskbar?
I don't know what you mean.
Okay, so there's no interference from some other computer taskbar coming up at the top, right?
No.
Okay, good. All right, so now we switch to some other application of the technology in the nutritional supplement industry. That's a multi-billion-dollar market, and there the increase of the carotenoids in the skin over time, here from day 1 to day 28 in some subjects, is interesting for them, because they can now use this technology as a sales tool and convince people that that their supplements work. So if they give supplements to some subject at day 1, show them that their skin carotenoid levels have increased, then maybe they can increase the supplement sales. So that's what we set out to do. And we, for this purpose, we developed a portable instrument. Another instrument which is based on a 473 nanometer solid state laser. It fits into this envelope here. This is the commercial instrument. And this is made possible by the invention of a solid state blue laser here, which operates in the infrared wavelength region then, which is doubled, frequency doubled, with the help of very strong LED light sources, which have also emerged in the last 10 years. And as a result, and in combination with fast computers now, you can develop portable instrumentation, which is kind of as big as a shoebox, not too big, and you can interface it with laptop computers. So very portable. Okay, here's the laser again. Okay, here's the latest development. In the meantime now, they were able to get rid of the laser and replace it with a filtered blue LED, which, as you know, has been very important in optical technology. Developments of very strong blue LED light sources. And if you filter them, they're strong enough to use them instead of a laser. So this has a blue LED laser in it. You see it's very compact here. You hold the palm of your hand in front of the light source, which comes out here. And then you can measure your carotenoid score. Here's shown the histogram for 33,000 people, which they have measured in the beginning. And you see a nice histogram here, which is slightly skewed. And they like it here because they can show that people who consume a lot of their products score higher than people who do not score, people who do not consume their products. Currently, they have about 15,000 instruments in use. You can do some demographics here by you can distinguish between age groups. For if you have these high numbers, and you see on average, it's pretty much independent of age. Just depends on the diet. Here's the scanner readings versus supplement usage. Goes up. The more supplements you take, the higher the readings, and so on. By the way, how are we doing here time-wise? I could go on for a while longer, or do you want me to continue tomorrow? Hello?
We have already gone over almost 1 hour and a half. We can stop here.
Okay, yeah, that's a good time to stop here, and then I'll continue tomorrow.
Okay. Let me ask if there's some questions, but only 3 more questions for Werner, for Dr. Gellermann. This time to end the talk. Pregunto si hay alguna ¿Alguien tiene alguna pregunta? Nada más, no más de 3 preguntas para terminar la sesión de hoy. Por favor, los que tengan preguntas. I have myself, I have one question. The technology, the optical technology you have developed, would it be possible to use in the detection of disease marker molecules, for example, in human breath, which is molecules in gaseous, in the gas form? I'm thinking about detecting ketone molecules in control and detection of diabetics, for example, from the human breath.
Yeah, that's a interesting question, but people ask us, can you also detect other molecules? And people are trying that, but so far there has been no success with optical methods. It's pretty much only the carotenoids. And the reason is that the Raman effect is very strong because of resonance enhancement. Normally the scattering is very weak. So you have very, very low signals requiring, excessive collection times of maybe half an hour or so or longer. And, since they're resonantly enhanced, we can do this only within about 15 or 20 seconds. So the answer is, unfortunately, it's very limited to specialized molecules here. And we are lucky that there is no fluorescence coming from these molecules. But people are trying to detect other molecules. For example, glucose. As you know, right now, they're still needed to prick the finger and take a little blood sample from the finger. And people would love to use optical methods there as well. And other molecules too. There's one possibility where you can detect oxygen saturation with the pulse oximeter with an optical method. You might have seen that where people have developed a skin clip. You clip the device onto your finger and you can measure optically the concentration of the oxygen in the blood, the oxyhemoglobin, and make sure that you have adequate levels. Actually, the latter was interesting recently because from COVID, it's known that the blood oxygenation drops very quickly. And so people have been buying these pulse oximeters quite a bit recently, including myself, to check that the blood oxygenation stays high. Okay. But overall, there are very few optical methods. I only know about these 3 now. Our Raman, maybe some blood oxygenation measurements, and maybe the beginning of glucose measurements, but nothing else really.
Okay, there's another question here in the chat. It says, I'm going to read it. Do you check any other bone apart from the femoral bone?
Yes, we did. The surgeon tells me that the bone marrow, which contains there's a certain type of bone which is called spongy bone, where we find those carotenoids, and he tells me that these carotenoids are actually existing all over the human skeleton. So they should be very important also for bone health in other areas. It's just that this person was very interested in some research, and he had the clinic and the samples and so on, but he tells me that it would also have some relevance for other areas of bone fractures, which where you could see improved healing times with good diet.
Okay. That was a question from Israel Arrieta González. ¿Alguna otra pregunta? Ya para terminar la última pregunta.
This mechanism is similar of the sensor, the sensors ultrasonics.
Ultrasonic sensors?
That this mechanism is similar of the sensors ultrasonics.
No, this is optics. It's based on the Raman scattering of light. That's a light scattering phenomenon. For ultrasonics, you deal with sound. You launch some sound wave at the tissue, and then you look at the sound coming back, the reflected sound. Here we use photons. So we use laser photons or other photons and look for the scattered light coming back.
Okay, thank you.
Okay, Werner, I think we stop here. We are very thankful for your talk of today's talk. And we ask the audience to stay tuned for tomorrow talk that Dr. Gellermann will continue to provide us. Pues aquí vamos a terminar entonces la plática del doctor Werner Gellermann y los esperamos para la próxima plática el día de mañana a la misma hora, a las 11, con el mismo liga que anunciada para la para las pláticas. Y entonces aquí agradecemos también a todos su interés y su presencia y pues nos estamos viendo hasta mañana. Thank you very much, Werner.
Okay, thanks, Jaime. I'll see you tomorrow.
We'll see you tomorrow. El administrador puede terminar de grabar, por favor. Bueno.
Gracias, nos vemos mañana. Hasta luego, doctor.
Hasta luego, Luis.
Esté bien.
Bye.
Bye.
Sí, por ahí, nada más te iba a saludar, pero se me vino encima el tiempo.
Sí.